Mm. Mm. Hi guys. Sorry, I didn't even realize you were actually on there. Just trying to set up my um stuff right now. So thank you for those who are joining me. Excuse my throat. I've got a bit of a cough today. What I'm going to do today is I want to look at paper two and try and cover some stuff that I haven't done for a while so that I'm making sure I'm hitting all the topic areas. So if you are joining me and you are doing OCRA and you're looking at okay, I'm gonna give me a second because I didn't answer your question last time, so yeah, give me a second, I'll find it quickly. Where am I going? Which paper did you say it was? Paper two, 18A. Let's have a look. Um, hmm, second, just get it. Okay, immobilized enzymes. So I'm going to cover a few different things today. Let me just do. Oh wow, my voice is really going to go. I'm going to have to be very careful with this. Right, question 80. I'm going to cover Amna's question to begin with that I didn't manage to do last week, but then I want to cover a bit on some questions to do with microcultures, and then I want to cover some other things I think potentially could come up. So let me just find the question again. What did you say? 18A and B. Right, 18A and B. Where are you? Would that be correct? Right, give me 1 second. Let me get this down here. Right, 18A. Right, state one other method for wow, this is really going, isn't it? Immobilizing the enzyme. So they've told you about the covalent bonding, they've told you about adsorption, and they've told you about putting it into a matrix and trapping it. 1 second. Just go here. Second, guys, I'm just setting up. It's not what I wanted to do. Indicator. I'm just setting up. Okay. Right, so if I move that over here, that therefore means that should be going live for people to. Right. Okay, back to this question. Sorry, I'm gonna give me 1 second. So you had said about ionic bonding. Oh, interesting. That's an interesting question because it's given you covalent bonding, adsorption, entrapment in the matrix. And obviously, when you look at the different methods, ionic would be linking it to, let me just bring up a slide for you. Where am I going? You're gonna have to bear with me today. My brain's not as on it as normal. Okay, so in this case, we would be looking at, where have you got here? So obviously you've got the enclosed, you've got the covalent, you've got the ionic bonding, you've got your adsorption, and you've got your entrapment. So technically, yes, they should give you that as a marking point because you haven't said anything that's wrong. And therefore, I would say that they would give it to you, but there's no guarantee. It's not on the mark scheme. So the fact it's not on the mark scheme makes me think that potentially they've given it as maybe extra information because there's no do not allow, there's no ignores. So I think there wouldn't have been that many students who actually wrote ionic bonding. So I would say that they would potentially give that to you. Hi Sahib, hi user. I left a comment on your video earlier, but I was wondering if you could explain the difference between vaccination as I didn't understand an exam question on it. Sure, give me 1 second and I'll do that one. Encapsulate trap. Yeah, so encapsulation is the right way to do it. They would allow that in that question. And then 1 second, let me just go into B and finish this one for Amna. And then I've got a few questions that I want to cover tonight for as long as my throat will allow me to go. So this next one then, right, one advantage of using, oh, that really did go. One advantage of using immobilized enzymes is the enzyme can be reused many times. However, the process of immobilization can reduce the activity of the enzyme. Okay, we know that enzymes, before I even move on to answer the next part of the question, the activity of the enzyme is dependent on the enzyme and the substrate forming an enzyme substrate complex. So therefore, the enzyme is a protein that has a tertiary structure. That tertiary structure has got a very specific shape. That specific shape is going to allow for that active site to be able to mold around. So I'm just putting out some notes in just fit. And because of that, that's therefore going to mean that it can work in being able to break down the substrate into the products. So explain why the activity of immobilized enzymes might be lower than that of enzymes that are free. So if they've been immobilized, right? So if you think about it, immobilization is sticking it into position. So I'm stuck on this chair. I'm immobilized. I can't move. Now I can move my hands like this. However, I can't move. Technically, I can because I'm on a chair that moves around. But if you think about being trapped in an area, because, thank you, because you can't actually move fully. So if you think about the protein has a tertiary structure. That's those interactions between those R groups and the fact that it can mold itself around the substrate, and that puts strain on the substrate, causing it to break apart by lowering the activation energy. So because of that, what you have to realize is, in this case, if we have them free floating, their shape is intact. Whereas if they are immobilized, their shape might be compromised just slightly. So that's where I would believe that they want to go on that one. There we go. Covalent bonding matrix carrier might affect the shape of the active site. Active site might be hidden when bonded to the carrier, and substrate must move through the matrix. Does that help, Amna, on how to answer those two questions? Do we need to be able to explain examples of natural cloning, like? Hold on 1 second, I've got to get my head around what you're saying there, Tony. Do we need to be able to explain OIR examples of natural cloning? You should know natural cloning in plants, absolutely. And you should know natural cloning in animals. So things like the tubers, the runners, excuse me, the bulbs, you should know about those. But with regards to questions, if they're going to give you a question like that, they'll probably give you some form of an example, would be my assumption. Could you please go over the difference between batch and continuous fermentation? Um, if I say your name wrong, I'm so sorry. But Cora, if you go to my live that I did yesterday, I want to say, I actually did batch and continuous fermentation in that live. I'm going to answer some questions on it here. That's my next thing I want to do. So I will do that. They have asked about bulbs and runners in the past. Yeah, they have. So you do have to have your known ones, but they could give you any examples. Yep, absolutely right. You don't need to know them. You just need to know about the fact that they can give you any example in the exam. And the key thing is that you know how to apply your knowledge to the example they've given you. So okay, right. So hold on, there was a question before, and it was actually on my list of ones to reply in videos. User, you asked about the difference between a vaccination and immunization. So sometimes those words can be interchangeable. But vaccination, when you talk about a vaccination, you're talking about the weakened or dead version of the pathogen that is going to be injected into someone. When you're talking about immunization, you're talking about the body's response and how it deals with the vaccine that's been put inside. So it really depends on the question about what have they actually asked? What do they actually want you to give them with regards to the information? And it all, in those types of questions, if they are asking for that difference between the two, it would be a case of that they've given you something. B cell, T cell, please, I always structure difference on what they do. Okay, if you are just joining me and you've never come to my lives before, what I would suggest for the B cell T cells is go over to my video that's specifically on the immune response. They're in module basis. And the reason being is I don't feel like that much is coming up now. Don't get me wrong, they could ask questions on it, but they did a lot on the T cells. So if they were going to do anything on the immunity side of things this year, it could be about antigen presenting cells, maybe. I don't know. I don't have a strong feeling about that coming up. But definitely go check out my video because I have videos on the whole course. But what I do want to do, just now, for as long as my voice actually lasts, I've got to be careful because I need to teach tomorrow, is just go through some questions about oh, excuse me, about the stuff I did last lesson. I say last lesson in yesterday's live to talk about the batch continuous and what that actually means. So this is very similar to the picture that I was showing you yesterday, and this is where you need to be thinking about the fact that what would see. So the question that was set up in this one was about this is a stirring rod, and that would be necessary for that even mixing, if you remember, to ensure that there's even distribution of the nutrients as well as the products that are being made, so the nutrients that are going in. A is where we're going to have oxygen being provided, and that's going to be needed for aerobic respiration. Now, one thing I will say to you is don't get caught up because respiration came in multiple choice, came up in paper one, that it won't come through again because respiration is everything basically. And then B, absolutely, is the cooling jacket. And that's to ensure that the temperature doesn't get too warm. So it was a multiple choice with various statements on vaccine. That's why I use it, yeah. So it's very much, they tend to use them interchangeable, but it's about one being about the dead or weakened version and the other about body's response. Okay. The type of question they may do in the exam, which is why I want to show this one here, is actually looking at if you have a graph, okay, so they've given us a graph and they've told us in this graph that this bacterium that they've grown is amino acid, which is lysine. Now, what they are wanting to do, how do you know that A is an oxygen supply? I know A must be to do with an oxygen supply because I know that microorganisms need oxygen to do aerobic respiration, and I'll show you as we go down with these questions. If it weren't being supplied with oxygen for aerobic respiration, then what would happen is it would start to be doing the anaerobic fermentation, and anaerobic fermentation is actually not a good process for fermentation because you get that buildup of ethanol, and the ethanol will therefore have a toxic effect on the microorganism that you're trying to grow. So therefore you want to aerate it to provide the necessary oxygen for aerobic respiration. Does that help? Okay, moving on to this question and carrying on with it. So we're being told we've got a graph that's showing the production of amino acids and the lysine. Now, it's important we can see here that my dry mass of my cells is increasing. So the fact that my dry mass of my cells is increasing, we're basically talking about my biomass. So my biomass is increasing. And what we can also see in the graph is that the relative concentration of the lysine is also increasing. So they've told me in the question that lysine is a primary metabolite. And because they've told me that, they're asking me, what's the evidence from the graph? Well, if you were in my session yesterday, you'll know that the primary metabolite is during the active growth phase. And so therefore, this is evident in this graph that there is an increase, and that increase that is happening is produced early in the cycle, and it's produced throughout the growth stage. Absolutely. Absolutely, Amna, because it's increasing and then it starts to plateau. And you can see that that increase before the plateau. So if we go down here, the increase before the plateau is following that growth that was happening. Let's try and do that. So we'll come down here, is following that growth that was happening before. So make sure when you're looking at these graphs, they're not going to give it in exactly the same way you've seen it before, okay? It's never going to be identical to what you've seen in textbooks. Some do actually. So a little bit I would say to you is have a look at the extra boxes. You'd be surprised how many of the graphs and stuff in the textbooks that they then reuse in the exam. So if it's like an extra, hold on. Here we go. If I just pull the book out here, so here is the OCR book. If I just grab, there we go. So I've just turned to page 170 and it talks about the histology of exchange surfaces. So for example, they may decide to use these pictures, like one of these pictures in the exam that they decide to do. The other one that they love to do, which comes up all the time, is where is it? Hold on, I've gone to the wrong place. I want photosynthesis. Where are you? Photosynthesis. Where are you? Where are you? Where are you? There you go. A similar graph. I don't know if you guys are going to be able to see this. If I put it on here. There, that graph there where it's showing you about GPTP and RUBP. You love doing that. Anyway, back to this one. So this next, what topic am I doing? Yeah, it's a bit rough, actually. Abdullah, you're absolutely right. Thank you. This is what happens. I always get laryngitis. I call it end of termitis, because I'm working so hard trying to provide for you guys as well as my private clients who pay for me to be able to do these things. So this topic that I'm doing is module 6, biotechnology, cloning and biotechnology. Right. Coming back to this question though, talking about how do the function of primary metabolites differ from those of secondary metabolites? So remember, primary metabolites are the ones which are needed for the active growth phase, cells metabolism. So if we're talking about glycolysis, so somebody thinks this is about respiration, and thank you, Amna, for letting them know. It's not at all. But this is where you'll get confused because this module, which is module 6, is actually looking at biotechnology. But how do you culture microorganisms if they can't do respiration? So this is why you need to understand, if we were looking at the carbon cycle, what's the carbon cycle involved in? It's involved in respiration and also photosynthesis. So these topic areas can come through again. Do not get hung up on the fact that they asked us loads of questions. We'll never get asked that in paper 2. It depends how they ask it. They're not going to go into the detail of the Krebs cycle or the Lynx reaction or any of the oxidative phosphorylation, but they are going to expect you to know if it's aerobic respiration, there will need to be oxygen provided. So the secondary metabolites, this is the important part of this question. It's that secondary metabolites. Now remember, as I said yesterday, that's during the stationary phase. So during the stationary phase, you're going to have a very high number of cells. So because you've got lots of cells, you're going to have lots of competition for those resources. So therefore, those secondary metabolites are going to be needed for giving advantage in that environment. So it is important that you note the secondary metabolites is for competition so that you've got an advantage. Because as the number of cells are growing, that's going to become a problem. Do you think it's too late to learn it all and get an A? Not if you work extremely hard and you've got no other exams. There is still time, but you need to focus it. Can you suggest any chance that do walkthroughs of past papers for OCR? I mean, I do questions as I go through it, so check out some of my lives. Amna's been amazing asking questions on most papers when I do these lives. So if you go back, I do different questions on different ones, but I don't know anyone who just goes through them from paper, each one right the way through, because it would take quite a long time. Right, hold on, let me see. Any predictions? Lots of predictions. That's basically what I've been covering when it comes to these topic areas. Gene regulation, definitely gene regulation. I feel like that's coming up. Whether it's transcription factors of or lack operon, I don't know, but it's definitely coming through. I feel like there's evolution coming through. I feel like you're going to have evolution classification, maybe linked to speciation. I definitely feel like your fermentation is coming through, batch and continuous fermentation. What else do I feel like's coming through? DNA. DNA is coming through so much because they haven't done anything. I think they may go down DNA replication, but I'm not sure on that one. But yeah, it's a hard guess, basically. Yeah, phylogeny, definitely phylogeny. Phylogeny linked to DNA sequencing, linked to population changes, so allele frequency changes, linked to oh, what else do I think it's linked to? See, this is where I'm like, oh, my brain is not functioning as much as it normally does. Yeah, linked to speciation, mutations, migrations, phylogeny. I feel like that's all going to be interlinked. I'll show you a question on that actually, because I do have it here. Is it easy to predict what's coming up? It's not easy to predict. I mean, I've seen paper 1, I've gone through paper 1, I've sat it. Quite interestingly, I've sat it as if I'm a student like you guys. And I will wait until the mark schemes are available to us as teachers, and then I will go through and I will see how many I will get right and how many I will get wrong. And just be aware, I've been teaching this for over 20 years. And when I say teaching for over 20 years, I've been teaching since I graduated from Cambridge with my postgrad. I've done lots of other things with regards to being head of biology and all that, but I've been teaching biology for at A level and IB level for 20 years. I came back to OCR A level 2 years ago, which is why I built this channel recently. I still make mistakes, even with 20 years experience and a degree and having got an AS equivalent in biology, I never get 100%. None of your teachers will. Okay. That's one of the things that anyone who tells you that they would get 100% in an exam if they sat it with you is someone very, very unique. I'm not saying it's not possible, but they're very unique. We all will make mistakes. That's why the grade boundaries are slightly different. And also, it comes down to mark schemes are based on how you guys do as you sit it right now. So the mark scheme are sitting there ready, waiting for you to finish these exams. But what they'll then do is they'll collect a sample of those papers. They'll mark them using that mark scheme, and then they'll adjust that mark scheme based on some things that people have answered, which aren't on the mark scheme, but actually they're like, do you know what? Yeah, we will allow that. Or, no, definitely don't allow that. And that's why you'll see the additional information that comes out that says allow, do not allow, etc., etc. So you will see that end mark scheme that you've been looking at for last year, but you don't see any of the extra bits the examiners get given. Us as teachers don't see any of the extra bits that the examiners are given, and the examiners are given it that year and that year only. And so therefore, that means that it will change every year. And that's why the mark schemes are never, this is exactly the answer they want. And only that will come up again and again and again. It won't. Every question changes. Every year changes. There was 20 odd thousand of students who sat it last year. You're never going to write it in exactly the same way. So stop freaking out if you don't know how to write it the most scientific way, and just try your best to answer it using a bit of common sense. If you're not sure, have a guess, use common sense. You might get a marking point. You'd be surprised how many times you can just by doing it through a bit of common sense. Do I think the grade boundaries are going to be lower? No, I do not. I feel like the grade boundaries are going to be exactly the same as they were last year overall. I think maybe 1 or 2%, but I don't think there's going to be that much of a difference. Right, sorry, I'm going to look at the TikTok questions now. Sorry, guys, I'm not trying to ignore you. Can you do the A-level cookbook does good ones? There we go. Charlie suggested the A-level cookbook ones. It's easy to predict what comes up. Were you confused by any of the questions like the Q10 one confusing? No, Charlie, when I say I wasn't confused, it wasn't that I was confused, but it took me time to process it. It took me time to figure out what it actually was asking me. So I did a lot of calculations, more than I needed to, just because I wanted to see. Do we need to memorize specific dates and agreements? No, don't worry about specific dates and agreements. If you've already mentioned it, but do you think enzymes are like enzymes that will come up absolutely 100%? Enzymes are involved in everything to do with biology. How they ask it will be different. So they ask quite a lot on the enzymes in your paper 1, but without enzymes, you wouldn't be able to do anything biological. So that's basically where I would say that one's going to. I'd say paper 1 was hard, but it wasn't any harder than anything I've seen in the past. I feel like it was very similar to 2024's on that level. Never memorize. You've got to be careful. You can't just memorize things. If you're memorizing things, you're not going to get the mark that you need. So this type of question, just to finish this off, industrial fermenters, they're given the glucose and the urea, and they need to make amino acids. So basically they're checking, do you really understand the structure of amino acids, the structure of urea, glucose, what's it needed for, ATP production through respiration. The ATP can then be used, and with the urea, we're going to have the amine group that's going to be made to make amino acids. But to be able to take the amine group away, you therefore need to be able to do that conversion. I wanted also, oh, excuse me, look at a question like this. They love throwing graphs at you. So one thing I would say to you, if you've got a graph, and actually, you had the limiting factor graph, you had the log scale on a graph? You had, I feel like this paper is going to ask you to draw a graph. So paper 2 will be to draw a graph. They're going to assess you on drawing a graph because they haven't done it yet. So if they don't do in paper 2, they're definitely doing it in paper 3, but I feel like it's coming in paper 2. They haven't done a lot on fisheries either, which I kind of feel like that may come through for population sustainability if it's not a random specific example. Um, okay, so looking at this, one thing I would say to you is make sure when you are doing this that you actually look at color coordinating. Fermenter, oh, that was a really dodgy line. Let's try that again. Fermentable sugar. Then you've got your yeast cells. Then you've got your ethanol. Then you've got your nitrogen source. Now, make sure if you do it like this, you know which scale you're using. So ethanol was green and yeast was blue. So try and color coordinate as you're going because that will help you to be able to see the different numbers that are involved. So whatever question they ask you, you are visually aware of which lines you want to look at, which ones you want to compare. So if they give you a question like this, explain why the yeast population stops increasing. Why is it stopping to increase? What's going on? What's happening that's going to stop it? And this is where they want you to understand, well, it's actually stopping at the same point when the ethanol concentrations got really high. And you should know that ethanol at high concentrations is toxic. Why is it toxic? Well, let's think about yeast. What is yeast? Yeast is a eukaryotic organism. And because it's eukaryotic, it's made up of eukaryotic cells. These cells have got a membrane, a plasma membrane. That plasma membrane is obviously made up of a phospholipid bilayer. They haven't done anything on phospholipid bilayers yet. Phospholipid bilayers. And because they haven't done anything on phospholipid bilayer, they could link that in here. So when you have the ethanol concentration increasing, the ethanol is going to disrupt the membrane and therefore kill the cell. It's not only going to disrupt the membrane, it could also disrupt the proteins from being able to function. So this is where you have to be able to link those topic areas together, but they do love throwing in a graph that looks like that. And then they may actually ask you to use the graph to then say at what time it would be most economical, because we actually want ethanol if we're trying to harvest the ethanol. So remember, fermentation is about doing this to get a product which is beneficial for us. So if we're using biotechnology, we're growing these microorganisms for producing the alcohol, brewing the beer, the pure alcohol, the vodka, the gin, the whatever spirits you want to have, we're going to want to harvest that ethanol at the right level that's not too high that it actually becomes a problem. So when you're looking at something like this, whereabouts would you actually want to harvest it? This is where they want you to think about when it starts to slow down. So between days 4 and 5, somewhere between here, we're going to want to harvest it. So no, it depends, Zach. Zach, it depends whether or not. So if we were doing The batch fermentation, we're probably going to stop that at the stationary phase and collect the secondary metabolite. But it really depends what they decide they want you to be looking for. So if they're talking about it in the, if we go back up a second, if we were looking at this previous question, if we wanted to actually get lysine, then that is a primary metabolite. So we would want to harvest the primary metabolite during the log phase, during the active growth. So therefore, we would want to use a continuous fermentation process so that we can maintain it at that exponential growth. If we're doing batch fermentation, however, we would want to ensure that we are getting it up to that stationary phase, so we're getting the secondary metabolite. And just because I've just spotted it as well, see, and this is one of the things I didn't even notice as I was doing this question. Hold on, where am I? I'm on that page. I want you to know, someone said to me, how did I know it would be aerobic? And how I knew it was aerobic and it was air, A, is it says it's an aerobic fermenter. Yes, Zach, absolutely. So it's not essential for the active growth, but it is something that is produced. Yes, it is available for afterwards, Zara. That's why I do them on the YouTube and on TikTok. So those of you who are on TikTok can have it as well. But those of you who are on my YouTube channel, you have access to these videos that you can watch them. You can skip past 2 points that you need to know, etc. So it's there to help you, and I hope you find it helpful. Okay, coming back to the question that we were just on, which was this one. At what time would it be most economical? Between days 4 and day 5, because actually after that you're plateauing off, so you're not going to get any more ethanol. So therefore, you want to harvest between days 4 and 5. So do make sure that when you're doing these sorts of questions, that you are looking at the information that's been given to you. Focus on like it was literally all respiration, photosynthesis. It wasn't, user. It wasn't. It was enzymes. It was hold on, let me think. I've got to remember because I did it the other day there and I'm like, my brain is frazzled. It wasn't just respiration and photosynthesis. It was about application skills, but it was the content through it, but you just had to know where to look. I feel like DNA, so module 2 will come through in all through. So module 2, DNA, I feel like is coming up. I feel like biological membranes is coming up because they haven't really touched on that much. And I feel like lipids is still coming. I've still got a strong feeling that lipids is coming and where they're going to bring it up. I'm not entirely sure. They came up in paper one could come up again. Yes, they can come up again, but they'll just ask it in a different way. Cellular control, absolutely, Fran. I'm sorry to say that for you, but cellular control is coming. I feel like it's coming. I feel like it's coming quite high. I do feel evolution, for those of you who weren't here earlier, evolution classification, I feel like is coming. But I feel like the way they're going to do it could be a bit bizarre because patterns of inheritance, natural selection, and Hardy-Weinberg is a great topic to assess your understanding of how things are passed on. And then if we look at it to do with the environment and how organisms are able to survive due to the selective pressures, they could ask it, but ask it in a way that's about the environmental factors and the biodiversity and how you can have the biodiversity changing because there's been a random mutation. That random mutation resulted in a variation which was then making one more advantageous than another. So again, it just depends which way they want to go. Biology is the bigger picture. They can ask all the topic areas in one question if they wanted to, which is really fascinating because there's so many students who go, Christine, how can you spend 45 minutes on one question? I'm like, well, because I can go any which way I want with that question because it's biology. It's all interlinked. Would they ask another statistical test question? Yes, they could. So I know they've already asked you your t-test question. I feel like there's maybe a Spearman's rank coming through because they did chi-squared last year, didn't they? They did chi-squared last year. They've now done a t-test. They could, but I don't think it'll be in the same way because this one you just had to use it to evaluate and work out your degrees of freedom. You didn't do the calculation. So they could bring in a t-test again and get you to do a calculation, or they could get you to do a standard deviation calculation. Yeah, they can. They can do a 6 marker on that one. I feel like there'll be one 6 marker, which is probably more to do with ecosystems and analyzing data, which I'm going to show you in a minute. It links this question. Now, this question's not from your exam board, but I like it because it links the two together where I feel like it's going to go, or it could go. So let me just go through this question and then I'll come back and start answering some questions for you. So here you have a picture that's talking about this tawny owl. You're being told some information. So let's just look at what they tell us. They've told us we've got feather colors and they can either be gray or they can be brown. We're told that they're controlled by a single gene locus, and we're told that the brown allele is dominant to the gray. So I therefore know in this one, because they've already told me a lot of information in the question, I know that I can say, right, I've got brown allele, which is dominant to my gray allele, and therefore I could have capital B, and I could have capital B, lowercase b, and I can have lowercase b, lowercase b. So I know about that. So now what I need to do is I need to say to myself, right, well, if I were to take two heterozygous and I cross them together, so let me just do a Punnett diagram on this one. So here's my Punnett diagram. So I'm imagining that I've taken an F2 generation. I therefore got two capital B's, a capital B, lowercase b, capital B, lowercase b, and then two lowercase b's. So what have I actually got? Let's just write this out. I've got capital B, plus I've got two lots of capital B, lowercase b, plus lowercase b, lowercase b. Well, Hardy-Weinberg is basically that, except for instead of doing capital and lowercase, we've got PP plus PQ plus QQ. We've got two of them, and that's what Hardy-Weinberg's equation is. So therefore, because that's Hardy-Weinberg's equation, we are able to work this out because they've told us 84 are gray. So 84 are my B, two lowercase b's. So therefore, that means that this will be in the lowercase one. So my Q squared equals 84 out of 120. So because I've got 84 out of 120, I therefore know that my Q squared equals 0.7. And then I need to work out what Q is. So Q is the square root of 0.7. So therefore, because I know Q is the square root of 0.7, I therefore know that Q is equal to 0.837. So now what I need to do, let me just rub out this Punnett diagram. Now what I'm going to do is I'm going to say, right, well, I know that P plus Q equals 1. So therefore, I've already worked out Q, 0.837 equals 1 if I add P to it. So if I move this to the other side, I'm going to minus 0.837 from my 1. And therefore, because I'm minusing 837 from my 1, I can note that my P is equal to 0.163. So now I need to look at the question. It says heterozygous. So I know heterozygous is 2pq. So therefore, 2pq equals 2 times 0. In fact, let me do it the right way around. 0.163 times 0.837, which equals a total of 0.27862. 0.27862. 27862. 62. Always make sure you go back and double check your answers as well. It's really important that you do that. So you know what it's saying. Right, hold on. Let me just come across because someone asked a question there. Right. Why isn't q squared equal to 84? Because it's 84 out of the whole population, and you were told the whole population was 120. So therefore, it's a proportion. Proportion means out of total, yes. Great. Potentially, if I don't get through it tonight, I'll go through it tomorrow. I will come back on. Well, I say that. If I'm okay, if I've got a voice by the end of tomorrow, because tomorrow I have got, what have I got? 6 hours of teaching. So if I've still got a voice tomorrow night, I'll be back on tomorrow night to do a bit more. And I will try to come back on Thursday night for at least an hour. So this is what I want to show you now. Oh, no, I didn't finish that one. Hold on. Hold on, I want to go back a second because I hadn't finished. I hadn't finished. So be careful. I just skipped ahead there really quickly. It actually asks me to calculate the number of owls. So what I need to do then is I need to take that number and I need to times that number by 120 again. And that will therefore give me the number, which is 32.7. And therefore, what I need to do is I need to either round down or round up. They'll actually accept both on that one. Thank you, Zach. I am very hardworking, but the thing is, I want to help as many of you as I can. So I want these resources to be out there, but also it's not just about you guys sitting these exams now, it's about future generations as well. I think about my daughters and I think about what about when they get up to A level? What about if they need some support? What is available to them? Immobilized enzymes hasn't come up recently. However, what was really fascinating with paper 1 is paper 1 talked about immobilizing the algal beads were immobilization, because you were immobilizing so that you could look at what was happening with photosynthesis. So I did have on my list immobilized enzymes as being a big question coming up, where they would use fermentation, the batch or continuous fermentation, to be able to get the enzymes that they would then immobilize, and they would then use them for some form of reaction. But whether or not it is going to go that way, I don't know. Okay, so now we can move on to this next question. And this next question is what I really want to look at, because this is the way, again, in which they can give you some information, and the information that they're giving you is in graph form. Yes, I would say potentially. In answer to how you would be able to control it, you want to have the mass of the organism. So yeah, you would want ideally to have the mass of the balls. So if you were doing the algal balls could be different sizes, but if you controlled the mass of the algal balls that were going into the hydrogen carbonate solution, that would therefore have an effect. But the one thing I would say is you can't change it. Don't stress about it. Move on and think about what you're doing now. Trying to lubricate this voice of mine so my vocal cords don't get completely destroyed. Okay, what have we been told? Climate change. How would climate change actually have an effect on the frequency of these two alleles? So this is really fascinating because a lot of you will have studied module 4 and looked at biodiversity. You will have studied module 4 and looked at evolution and natural selection, and the fact that you can have adaptations which are anatomical, physiological, and you should know that climate change can have an effect on the biodiversity. So in this question, it's really fascinating where they're asking about climate change and about the frequency of these alleles, linking it to patterns of inheritance and how the environmental selective pressure is going to have an effect on the organisms that are present and therefore on the frequency. Is it directional natural selection? Is it disruptive natural selection? Or is it stabilizing natural selection? So it's really important that you have gone over natural selection and you know the different patterns there are and how they could interlink it to an example, which in this case is about owls. Thank you, Amna. What would I do without you, by the way? You're like my little go-to person to help. Thank you so much. Okay, so coming back to this question, then let's have a look at what they're asking. They've asked us, right, this is a different exam board, just to be clear. So they've said, analyze the data. If this was an OCR one, they would say, evaluate the data, and that would be it. Not analyze the data, just evaluate. So they've already got evaluate there. Evaluate this conclusion using the data provided, okay? Oh, Anna knows who I'm talking about. Anna, even though there's more than one Anna, Anna does actually know what I'm talking about. Anna and I go back at many of these sessions. There's a reason when you watch my videos, in the fact that there are lots of questions that I've gone through because Amna's been doing the work for you guys. I mean, like, can you just explain this one? Can you do a bit of this? And that's what you need to do. So if you're a year 12 watching this, or if you are someone in year 11 thinking about doing A-level biology, start asking questions. That's how you will learn. Honestly, I know those of you sitting your exam tomorrow or literally on Friday, sorry, are like, it's so late out in the day. How am I only just hearing this now? But actually, it does make a big difference. So yeah, ask the questions because that's how you learn. And don't be afraid to ask the questions because you may think you look silly around with everybody else around you. But I tell you what, they're sitting there going, what? Employ her. I wish, I wish I was making money that I could employ her. I literally make the bare minimum that I could make to be able to still survive. So yeah, I don't earn on these at all, at all. So just to be aware. Exactly. Amna's right as well. Just make sure that you are understanding where you're going wrong. But I do want to talk about this question because this is a fascinating one because it's talking about the depth of snow. It's talking about the fact that, remember, there was different colors, gray and what was the other color? There was the gray and the brown one. And it talks about the fact that you're looking at the percentage that were colored brown that were going to be recorded. Well, why are they being colored brown? Well, what's happening? Climate change, what could that be linked to? Well, if there is too much, let's say CO2 in the atmosphere, why would there be too much CO2 in the atmosphere? Because of deforestation. If we're not doing it in a sustainable way, if we're cutting down our trees, they're not able to remove the carbon dioxide. They're not able to fix the carbon dioxide by photosynthesis. Therefore, carbon dioxide is remaining in the atmosphere, and that could be causing temperatures to rise. So in this case, if we've got temperatures, I get nothing on TikTok either. I don't get paid on TikTok. anyone who has gifted me on TikTok, which has been lovely, by the way, and thank you. At the moment, since I started doing my lives, I have got 40 pence. So yeah, it doesn't come through TikTok. I wish it did. You have to have 10,000 followers on TikTok to get into the Creators Fund. And then they select on what they allow you to have. I don't have any of that. That's not why I do this. I do this so that I can help you guys. And obviously, I hope that I'll start earning more money. For those of you who've bought my workbooks, amazing, thank you. That's the sort of thing that brings the money in. For those who are my private clients, that's what brings the money in for me. But these sessions I don't get paid for. So yeah, it's all about just me being helpful. And I enjoy it. I love teaching and I want to help as many of you as I can because I wish I had that help. Um, yeah, I'm talking about live streams right now, not about this question. Let's come back to the question. Um, so with regards to this question, so if we're talking about carbon dioxide, we're talking about deforestation, we're not removing the carbon dioxide, we're therefore getting a temperature rise. Not only will that result in a temperature rise, because I've got a feeling they're going to bring the carbon cycle with climate change, it could also cause the oceans to become acidic, linking you to carbon dioxide reacting with water when it dissolves. So you've got that as a factor. But in this case, what we've got is a temperature rise. Temperature rise has resulted in the snow, obviously the snow depth decreasing. Okay, there's less snowfall. Why is there less snowfall? Oh, well, hold on a minute. Where does snow come from? Snow comes from when the temperatures are low, and the temperatures being low, there being snow falling. Well, that's the rain, the precipitation that comes from transpiration, the water vapor getting into the atmosphere. So again, there's a big link around to how it could link to module 3 if we're on paper 3. But we're looking at genetics here. We're looking at how this can have an effect. So when you look at this, you can quite clearly see that there is a negative correlation. So there's a negative trend here. We've got negative correlation. It's going down because I can see that with my line. So I've got my grey owl trend line, and I've got my brown owl trend line. However, evaluate the conclusion. The increased human activity are resulting in an increase in the frequency of brown owls. So how are we actually linking to that? Well, you've got to be aware the evaluate is pros and cons. Support that conclusion, does not support that conclusion. So if we can say there is increase in our what am I trying to say here? If there is an increase in the owls, whichever color the owls are, that could be a selective advantage, which means that they are having that selective pressure. They're able to survive. Because they're able to survive, they can reproduce, they can pass on their alleles. So that's how it would be linking it to natural selection. So if we're looking here, we can see as the snow depth increases, there is a decrease in the brown owls and a decrease in the grey owls. So as the snow depth increases, the percentage of owls that are surviving decreases too. Excuse me. Carbon cycle is module 6, ecosystems. Yes, you can. Hold on, guys. I've just got to jump over to TikTok quickly. I have to move my puzzle piece. There we go. I'll move you out of the way. Oh, right. So one of the things to be aware of is looking at what they're saying as well with the data they provided you with. Zero data. There is zero data there. At 20 centimeters, we do not have any data. So because we don't have any data, we therefore need to understand that there being no data means, why is that not showing? Is it showing? Hold on. Yeah, it's okay. There is no data at 20 centimeters, so we can't be sure on this conclusion because we don't have enough data. The other thing to know, if we carry on, what's happening in the years, what we've got is we've got anomalous results. We've got anomalous results which are all over the place, and we have got anomalous results which are not only high but also quite low. So it's important to note that when you're looking at these sets of data, although you can see a trend, there is a lot of variability in this data. So because there's lots of variability in this data, that therefore means that that would not support. However, what you can see is there is a trend over here. So that would support, although we do still have some anomalous results which don't quite kind of fit our pattern of our data. So it's really important that when you're going through these types of questions, that you're looking at the data that's been given to you and really dissecting it. That's what's important. Right, let me just keep doing that. Have a quick look if there's any. Yeah, absolutely. Go over the stuff you hate the most. Because that's the bits that you don't have in your head. You will be surprised how much you actually have in your head. You just have forgotten that you know it. And so therefore you need a way of just pulling it forward. You may get a peat bog. I actually do. Amna would agree with you on that one. If they're going to give you an ecosystems question linked to climate change, linked to the effects of carbon dioxide, that could be in the example of a peat bog, and understanding that there is management techniques needed. So in peat bogs, they need to have the anaerobic conditions. But if the acidity gets too low, that's going to have an effect on the, enzymes being able to work. And because of that, that will then have an effect on the rate of respiration, etc. So I do feel like that could come up. Always, if you've got nothing else to say, absolutely throw in correlation does not imply causation, because that just is a it's like a fail safe, but it doesn't guarantee you a mark, just to be aware. Students tend to think that they can throw off these curveballs that will give them the marking point. Curveballs, not what I mean. You can put this answer out there and you'll get the marking point. Make sure you put it relating to the question. Just because there is a correlation between the percentage of owls that are brown increasing, that does not mean that the snow depth has decreased. Because there is a correlation does not mean causation, but link it to the question will get you the marking point. How do you work out your degrees of freedom? If it's a t-test, it's n minus 1, n minus 1. So basically 2n minus 2. So I think it was 8 and 8 minus 2. So you would have had 14, I would have said on that one. But if it is looking like someone asked about Spearman's rank, it's the number of values you have. So your sample, how many are you comparing? So if I've got 7, let's say, in this case, how many do I have? Hold on. 1, 2, 3, 4, 5. Let's say I've got 5 snow depths. Therefore, my sample size would be 5. That would therefore mean that my degrees of freedom is not called degrees of freedom, but not, because it's called sample size. So it's n, which would be 5. Yes, this is OCR. In paper 1, did you need to do the t-test and null to get 6 marks? Did you need to do the t-test? No. Did you need to talk about the t-test? Yes, you were given the calculated value for the t-test. You had to use that to come up with an answer. Um, and then what else? What time are we on? Guys, I am gonna finish for tonight because it's past my bedtime. Um, there you go, Amina. I am going to finish for tonight. Not Spearman's rank. No, Spearman's rank is just N. Hold on. So hold on, hold on, hold on, hold on, hold on, hold on. Let me just check this. Spearman's rank, Spearman's rank, Spearman's rank, Spearman's rank, where are you? Spearman's parity test. Spearman's rank. Yeah, so hold on. So this is, I'm just going to copy this straight across. Give me 1 second before I finish. So this is from the OCR's version, which is, hold on, where am I here? So when you talk about Spearman's rank, and this is specifically from their booklet, which is about math skills in biology, as it says here, you're looking at your case is N is the number of paired data. So therefore it would be 7. So if you're doing a Spearman's rank, then you would want to do it as, in my case, I said there was five paired values, so therefore it'd be 5. So if you're looking at your chi-squared, hold on, let me just put do 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 do. So if we're looking at the paired t-test, it's n minus 1. So paired t-test would be n minus 1. If we're looking at the t-test, which is unpaired, that one would be, and I'm double checking this against the information that I have because I'm feeling tired. I don't want to give you the wrong information. So I'm just going and double checking against this, which is what I would advise you to do, is always look at, yeah, NA, I was just double checking that, is, oh, crumbs. So you've got NA plus NB minus 2, that should say, not 1. And if you were doing the degrees of freedom for your chi-squared test, where have you gone? Chi-squared test is the number, degrees of freedom is the number of categories. Just put this in here. For the chi-squared, this is chi-squared. The number of degrees of freedom is 1 less than the number, oh, I totally missed out a bit of categories. So if, for example, you had, let's say you had a predicted ratio and you had a 1:1:1:1 ratio, you have 4 categories. Because you've got 4 categories, your degrees of freedom would be 3. So I hope that helps. Guys, I am going to finish for tonight. Please do, if you're just joining me now, please do go and check the resources that I've got on my channel. If they're on my YouTube channel, they're also on my TikTok, so whichever one you like best. But in the YouTube channel, they're actually under specification playlist, so you can look at specific topic areas and do the revision from there. So do go and use them, and yeah, have a good sleep. because you do need to also sleep. Have some rest. For those of you who've got math tomorrow, good luck. Yeah, I'll try and be back on here tomorrow night. I don't know how long I can go because it depends um how my voice does after my teaching. But yeah, I'll try. If I can't come on tomorrow, I'll definitely try and come on Thursday night at the end before the exam the next day. But thanks guys for joining me and have a good evening.